Welcome to our first presentation on flow cytometry. In this unit, we will discuss the components of a flow cytometer. We will also cover general principles of flow cytometry and look at some cases in hematology to appreciate its application. At the end of each section are some questions that will help you to review important concepts in that section. Please take some time to satisfy yourselves that you understand each section. You may do so by pausing the video and going back to the content you wish to review. As the name suggests, flow cytometry means the study of or measurement of cells. That's where cytometry comes from, using some fluids as they flow through the instrument. This flow part of the instrument is called the fluidics. Other parts of the flow cytometer are the optics and the electronics. Here is a schematic representation of a flow cytometer. The fluid that carries cells in a flow cytometer is called the sheath fluid. In this particular flow cytometer, the sheath fluid is propelled through the fluidics by applying air pressure into the sheath fluid container. The air is filtered to remove any particles that may interfere with the function of the instrument. The sheath regulator controls the amount of pressure entering the sheath container. The flowing sheet is then passed through the air filter to make sure no debris enters the flow cell. The flow cell is the happening place, if you wish, and we will discuss that in detail shortly. Here in the flow cell, the sample is introduced into the sheath fluid. The cells interact with a laser beam at the interrogation point in the flow cell before being passed to a waste container. The fluidics also include the sample movement. Air pressure is also applied into the sample container, forcing the sample into the sample injection tube and into the sheath fluid. The amount of air pressure applied to the sample determines the amount of sample and therefore the number of cells that will go through the flow cell. So, if the sample is dilute, you can increase the flow to capture more cells, but you can also decrease the sample flow rate to reduce the crowding in the flow cell. Slide 3 helps us to look at a closer look at the flow cell. In this flow cell diagram, the sheath fluid is shown in blue. The sample is injected into the center of the sheath, so the sheath fluid surrounds the sample. The sample is in pink, and there are 3 laser lights crossing the flow cell. These are the red, blue, and violet laser lights. The flow cell is shaped into a cone, and that has the effect of tapering the sample too, so that the cells pass through the interrogation point in a single file, that is, one at a time. This is called hydrodynamic focusing. The flow cell on the left shows good cell flow, while the one on the right shows too many cells crossing the laser lights at the same time. The result of too many coincidental passages creates the risk of counting 2 or more cells as one and can lead to a totally different interpretation. This concludes our discussion of the fluidics. Here are some questions to help you grasp the main points. And you may wish to pause the video here so that you can respond to these questions and satisfy yourself that indeed you understand what is involved. We now turn to a very important part of flow cytometry that deals with light scatter. The point where the laser beam intercepts the cell is called the interrogation point. As cells pass through the laser light, the light hitting the cell is scattered by the uneven surface of the interior of the cells. If the cell contains some fluorescent dye, it will also fluoresce, and the light will also be captured as additional side scatter. The light coming from the laser light is called incident light, and this will give rise to reflected light in all directions once it intercepts a cell. There are two sets of light detectors, the forward detector and the side detector, situated at right angles from the incident light. When no cell is passing through the flow cell, an obscuration bar obscures or prevents the light from reaching the forward detector. This obscuration bar is shown as a black line in this diagram. However, in the presence of the cell, the light is scattered in the forward direction around the obscuration bar to reach the forward light detector. The light then activates the detector, which converts that light to a voltage and registers a count. The amount of forward scatter is proportional to the size of the cell, so the bigger the cell, the more FSC or forward scatter. At the same time, the light is scattered to the side, and thus side scatter, also referred to as SSC, is measured. Side scatter measures cell complexity. Complex cells include neutrophils with segmented nuclei and granules. So, using the amount of scattered light only, we can classify cells according to their size and internal complexity. This is the basis for a differential count using a flow cytometer. Please notice that forward and side scatter is measured without use of fluorescence. The differential count is achieved without the use of any fluorescence whatsoever. In this diagram, forward scatter is measured on the horizontal axis, while side scatter is measured on the vertical axis. As cells pass through the flow cell, they are characterized according to their light scattering properties. Red blood cells and debris have small size and therefore have low forward scatter, around 50 channels. They are also low in side scatter, less than 50 channels. This allows us to separate the next set of cells, the lymphocytes, that are only distinguishable from debris by their size. The monocytes are larger than the lymphocytes and also have a folded nucleus and some granules that contribute to a lot of side scatter. This puts them a bit higher on the side scatter compared to lymphocytes. The largest and most granular cells are the neutrophils and eosinophils that show both high FSC and SSC. So this is how a normal blood picture looks like in flow cytometry. This diagram, I think, gives us a very graphic presentation of the cells. You may pause the video here to view some of the content, to review some of the content while it is still fresh in your mind. And these are the questions that you put up there to help you to review that content. Now we move to another aspect of flow cytometry, the optics. The optics of a flow cytometer is a system of lenses, mirrors, filters that direct light of a particular wavelength to a designated detector. For example, if we want this blue light to be measured on detector number 4, we will set up filters, lenses, and mirrors that direct blue light to that detector only. Here is the light, the incident light. Here is the interrogation point. Here is the obscuration bar, and beyond that is the forward detector. On the side detector, the light is concentrated by this convex lens, and the next filter does not allow it to pass, but reflects all of it into the designated detector. These filters are of different types. So we'll talk about types of filters. The management of light or optics in flow cytometry is achieved by the use of filters. There are many types of filters, but here we discuss 3 types. In the diagram above, the long pass filter or LP750 allows only light of wavelengths longer than 750 nanometers to pass. In contrast, a short pass filter, SP500, allows wavelengths of less than 500 nanometers to pass through. Any wavelength longer than 500 nanometers will be obstructed. A band pass, on the other hand, allows only light of a certain band of wavelengths to pass through. The band here is 50 nanometers as shown by that slash, which means 25 nanometers below and 25 nanometers above 525, or simply from 475 to 525. This is a little tricky, so make sure that you get it. You are sure to get this question one way or another. This concludes our discussion of Cytometry 1, and I hope you'll find these notes helpful.